Welcome and thank you for joining us for the second Agro Lunch and Learn webinar of 2024. This webinar series is only possible through the generous support from Eurofins AgroScience Services, and we thank Eurofins very much for their continuing sponsorship. My name is Solito Sumalong of the Agro Lunch and Learn Webinar Committee, of the Agro Division Strategic Planning Committee, and of AgriAuthority here in Kansas City. I'll be serving as moderator for today's webinar. Attendees should use the Q&A tool, we show you on the bottom, to submit their questions and comments, which will be answered at the end of the presentation. Now, before I introduce our speaker, I'll highlight the benefits of becoming an Agro member. Agro pays enormous benefits by enhancing your science, building your scientific network, and supporting your professional development. Please add the Agro division to your ACS membership, or go to agrodiv.org and click on Become a Member to become an affiliate for $20 a year, regardless of your membership status with the American Chemical Society. I'd also like to inform you about the upcoming ACS Fall National Meeting in Denver this August. The abstract submission deadline has indeed passed, and we expect to inform presenters of acceptances in May. There are also opportunities to apply for student and early PI travel awards. If you have any questions about programming, contact our program chair, Dr. James Foster. See agrodiv.org for more details. Now, I'm proud to introduce today's speaker. Dr. Charles Nyamekye joined Smithers as a senior chemist and study director. He directs studies and develops methods for product chemistry on a vast variety of test substances ranging from agricultural products to pharmaceutical drugs, with over 10 years of experience in analytical chemistry that spans across previous work at Iowa State University of Science and Technology and the Department of Energy at the Ames Laboratory. So, with no further ado, I'll hand it over to Charles.
All right. Thank you very much, Solito, for the introduction. Good morning, good afternoon, and good evening to all of our viewers. Thank you very much for joining us today, and I'm excited to be here to talk about the topic that has really been central to our work at Smithers. There's been a great deal of opportunity that has been on the rise for global regulatory prerequisite to support registration of products, and that has opened up new market for manufacturers called 5-batch preliminary analysis. I hope that by the end of the webinar today, we have been able to share and highlight some of the strategies developed and lessons learned to overcome the challenges facing registering different types of products to the global regulatory bodies. Now, as a quick introduction, a 5-batch analysis study must comply with the GLP principles of OECD, US EPA, the EC, and the FDA. 5-batch preliminary testing follows guidance from the Office of Chemical Safety and Pollution Prevention guideline under the 830-1700 series, along with the 830-1800 series. And these two guidelines are supported by enforcement methods outlined in the SANCO 3030-99 Revision 5 guidance. Now, after almost 20 years, this guideline is still being interpreted by our clients and manufacturers in producing their material. These interpretations have continued to evolve even through today. So 5-batch testing is defined by the US EPA as the identification and quantification of the technical grade active ingredient and the impurities from 5 separate batches in a production, in which at least 98% of the total composition along with their impurities has to be at least 98%. Now, impurities of significance or relevance has to be monitored at a level of less than or equal to 0.1% weight per weight. Now, it is not fully mentioned nor stated in the OCSPP guideline on what method validation guidelines are needed to support a 5-batch testing. However, we will highlight the changes that comes with our Sanko Revision 4 and Revision 5 guidance in the later slides. So we'll like to first have an icebreaker question to our audience. The question is, does your company or companies you work with require 5-batch testing? Always, frequently, rarely, or never? So we'll give you some time to answer our poll question today.
All right, I'll give the audience about 30 seconds after our lunch, which is right about now. Uh, 66% participation. I'm going to close that in 4, 3, 2, 1.
All right, so based on our poll questions, it looks like always and frequently we're at the highest with a split decision of about 36 to 38%. So thank you very much for addressing our poll question today. Now, for today's topics of discussions, we will talk about the significance of the 5-batch testing, our Smithers 5-batch cycle for success, analytical methods and instrumentations needed to successfully complete a 5-batch analysis, and lastly, challenges and lessons of a 5-batch analysis on naturally derived products. By naturally derived products, we mean plant extracts, chemochemicals such as hormones, biopesticides, and/or proteins and peptides. And these natural products would still need control of the makeup by the traditional 5-batch analysis. Now, 5-batch analysis is needed basically and is conducted on generic actives. Whether you are registering a plant production product, a biopesticide, or a generic active product, this registration still falls under the EPA, FARA, or TSCA registration. As a result, you may be required to verify the contents of the technical grade active ingredient produced by a manufacturer. Now, the 5-batch analysis allows us to assign all the toxicology data to the expected composition of a TGAI. Now, this becomes the reference source for determining equivalence of future batches. It is interesting to note, however, that in Europe, the TGAI employed in ecotoxicology registration should be characterized as you would in a 5-batch study. Now, this becomes extremely important because if the manufacturing process of your product has changed over time, such as changing manufacturers or when the production is switched from a pilot scale to an industrial scale for commercial use, the latter is considered a different source. And lastly, when the processing method of the manufacturer has changed and/or the use of multiple locations to produce that product, that also will require you to perform a 5-batch analysis. Now, it's interesting to note that if for a natural product, let's say the composition of the active ingredient is about 50% weight per weight, the other components to identify and ensure that the product meets regulatory requirements will need to be assessed. And these relevant impurities or minor components will be characterized and analyzed at levels that are greater than or equal to 0.1 weight per weight or lower for toxic and relevant impurities. It is also interesting to note that the higher the purity and the higher the clarity of the manufacturing process, the quicker and simpler a 5-batch analysis will be. In all, Smithers will help you develop the analytical strategy that is needed to identify and quantify active ingredients and significant and/or relevant impurities down to a level of 0.1% or lower. So for our Smithers 5-batch cycle for success, the main question that we ask our clients prior to conducting a 5-batch experiment is whether this new product that they're looking for to register on the market is an existing product or it's a new product. However, the manufacturing processes has changed over time. Knowing this information will gear us to determine the level of complexity and the challenges that we'll face in a 5-batch testing. So within the Smithers 5-batch cycle for success, there are 5 steps that we follow in order to successfully complete a 5-batch experiment. As I mentioned before, we'll review the manufacturer challenges, and then we will screen a relative batch. Whether this screening for an existing product is basically to screen for specific changes in that material, or for a new product to determine and to identify the impurities that are within it. Next, we'll verify and develop methods to quantify those impurities that are above our threshold of 0.1% weight per weight. Once we have developed methods for the active ingredient and impurities, then we'll use our SANCO 3030-99 revision 5 enforcement method to validate that method. And lastly, we'll perform a 5-batch testing based on the validated method. Now, there are numerous analytical instrumentations at our disposal at Smithers. We're able to obtain chemical information via FTIR or GC-MSD, quantitation analysis via ICP-MS, which can detect most of the periodic table of elements, specifically metals in the products, can also be employed. Also, due to the level of sensitivity, we're able to tune our analysis to meet specific detection limits of our test substance. For example, an HPLC DAD analysis versus an LC-MSMS analysis. Now, for volatile test substances that contain volatile residual solvents, we can analyze it by a gas chromatography flame ionization detection, or GCFID. If the compound is highly volatile, we have the capability of using a headspace gas chromatography mass spec to quantify the residual solvent and/or the impurities in the test item. Lastly, for test substance confirmation, we can utilize liquid chromatography, electrospray quadrupole time of flight mass spectrometry to determine the accurate mass and confirm the true identification of our analyte. Now, all of these instruments and analytical data, when combined together, can provide us the fingerprint and profile of any test substance, and it will help us successfully conduct a 5-batch study. Now, the most important and critical part of a 5-batch analysis is the initial purity screening. The primary key is the preliminary screen is a key component in overcoming the challenges that are associated with a 5-batch testing. Specifically, if it reveals unknown or unexpected levels of an impurity, which is deemed to be at or greater than 0.1% weight per weight, further analysis is to be conducted. Now, the initial screening and identification will progress us to obtain an efficient 5-batch analysis study, but it can also support our clients in selecting a suitable manufacturer to develop their product to meet these specifications and the regulatory requirements prior to conducting the actual 5-batch study. Most of the time, the results can lead us to more than one probable impurity or structure. Therefore, we may use various instrumentations to then confirm and identify that impurity. So in this first example, we show a manufacturer technical grade synthetic material with a known manufacturing process, and the impurity identification is not a blind process with infinite possibilities. Theoretically, they should come from the raw material, intermediates, and/or byproducts. Therefore, the initial screen for a representative batch of this material is shown here, and the analysis was conducted by HPLC DAD method with these conditions outlined. Now, an HPLC DAD method separates and characterizes the chemical mixtures based on the chemical and physical properties of each specific analyte. HPLC DAD does not provide us with a chemical footprint of these impurities. However, we can determine that impurities that are UV or visible active can be obtained with a light absorbing signal, in this case, our wavelength of 220 nanometers. Now, this is typically a percent area run for unknown impurities. However, for the definitive 5-batch testing, it is best to have impurity standards. And so in this chromatogram at the top right-hand corner, we show an acetonitrile blank, and therefore we show the signal here from our sample. From an initial glance and assessment of this material, there are numerous potential impurities that we observed across the retention time range. Now, these impurities may be at or below 0.1% weight per weight and may also be significant or relevant to our client. Therefore, with these initial screening, we'll always communicate our results with our client to ensure a successful GLP 5-batch analysis. Shown here, we have identified the active ingredient by our green arrow, and all of the impurities are highlighted by our red arrows shown here. Now, by using a percent error run analysis and a rough estimate of the percent error of each of the analyte peaks, we show our results in 5 representative batches. As you can see, impurities 2 and 8 have levels that are near or above the regulatory threshold of 0.1% weight per weight. This criteria is required by the guideline. However, we typically attempt to quantify at a level of 0.05% for impurities, specifically during these screening assessments. Now, the relevant and significant impurities is determined by hazardous versus active ingredient. It could also be something that adversely affects the stability of our technical grade active ingredient or is insoluble in preparation. However, these relevant impurities have to be controlled to prevent any type of adverse effects. Now, in our second example, we show a batch and a GC-MS analysis of a natural product. This natural product is an essential oil which was extracted from a plant. Our analysis was performed using gas chromatography mass spectroscopy detection with an electron ion source. Our analytical method, as well as the gas chromatography column, was performed by a dB wax column. Now, gas chromatography separates the components of a mixture in time, and the mass spectrometer then provides information that helps us aid in the structural identification of each component. Now, gas chromatography mass spec detection helps us identify these components within the natural product, and by utilizing the NIST library match, we can determine what those minor components or major components are within this material. Now, standard columns such as dB wax and dB624 columns are heavily employed at Smithers. However, we do have a variety of column packing that we can use for analysis. A dB624 column gives good separation of low molecular volatile materials. In contrast, a dB wax column is best suited for food, flavor, or fragrance type of applications. Now, keep in mind that some of the impurities within the natural product may have isomers and exhibit chirality. Therefore, the use of chiral columns with a cyclodextrin packing, such as a CP carousel dex CB column, can help us separate chiral compounds. Now, the challenge that rises with quantification of natural products within a 5-batch study is the unavailability of reference materials for those impurity characterizations and quantification. Now, this can provide us with an opportunity to use fraction collection techniques to isolate and purify the impurity of interest to serve as the reference material. Furthermore, we can contract synthesis from a third-party vendor, but this will be very costly. For reference standards that are merely not available due to the nature of the natural product, we will then seek alternative routes such as a percent error run to estimate the concentration of the impurity. Now, as you can see in this chromatogram, there are many components in the natural mixture that can vary from batch to batch, and it can be very challenging to identify or even have multiple matches of that minor component based on the NIST library match. Therefore, it is essential that we screen all 5 batches within a 5-batch study, not just one representative batch, due to the variations of the minor and major components. Also, we can utilize various instrumentations to accurately determine and identify minor components. Overall, extensive communications with our clients will be conducted to determine a proper, efficient, and cost-saving path to successfully complete a 5-batch analysis. Now, for this similar natural product, we have changed the method of detection. Therefore, we have used the flame ionization detector to analyze the essential oil from a plant. As you can see on the previous slide, compared to this slide, all of the minor components are not as sensitive due to the detection of our FID as opposed to the mass spectrometer. Now, gas chromatography flame ionization detector is versatile, reliable, and easy to use. It is mainly used to determine the quantity of molecules, unlike a GCMS, which can identify and determine the quantity of molecules of interest. The FID detector provides a pseudo response in which only about 1 in 100,000 carbon atoms produces that ion, but the ion production is strictly proportional to the number of carbon atoms entering the flame. Now, based on our signal from our GCFID, it is wise then to use this methodology and detection to quantify the major components in the natural product, while we use our GCMS analysis to quantify the minor components which are less sensitive in this type of detection. In all, we have identified 19 impurities in this natural product. After careful and meticulous evaluation of the data, we have determined that 10 impurities were deemed at or above the 0.1% weight per weight threshold. And that is indicated here by the pink line that spans across each batch. Now, a quick percent error run analysis was conducted to estimate these results. Therefore, these were not conducted using an external reference standard. Now, due to the high chemical diversity of this natural product, most of these levels are significant and relevant since they're at or above our threshold set by the EPA. As a result, the impurities will need to be validated under the SANCO 3030-99 Revision 5 guideline. Even though these impurities are above the criteria, they may still be registered due to the use of the natural product and its effect to our environment, and mostly its effect to us as humans. From this initial screening, we see that the variety from batch to batch of these impurities are problematic. There are many components in the natural mixture which can vary. As a result, it is challenging to identify or even have multiple matches of the minor components based on the NIST library match. Overall, natural products are difficult to analyze. However, by incorporating various analytical techniques and method, we are able to provide the fingerprint that our client will need to then determine how to move forward with a 5-batch testing. In our next example, we call this sort of like a knuckleball in reference to the most difficult pitch for a pitcher to throw in baseball. It is a knuckleball because we were not provided with any information about the nature of this biopesticide. All we know is that the biopesticide will be used in a fermentation type of product. As a result, we analyzed this material using our gas chromatography mass spec detector with these conditions listed here. We employed a DB5 column as opposed to a DB wax or a DB624, mainly due to the properties of this test material. Now, this is a major challenge, not knowing what to expect in a biopesticide type of material, in which we do not know what impurities to expect. However, the mass spectra of our MS can identify the mass spectrum of key impurities within this chromatogram, and that is from utilizing our NIST library match and the mass spectra shown here are 4 representative impurities. Now, despite not knowing anything about this biopesticide, we can start to generate the chemical profile of potential analytes. Also, different types of method will need to be employed to generate an overall profile of this biopesticide, such as LCMS time of flight to determine the accurate mass and confirm the identity of the analytes within this biopesticide. Therefore, the use of various analytical instrumentations will be at our disposal here at Smithers and will help our client to identify each of these impurities within the biopesticide material. Overall, we have tabulated our results. We found about 15 fatty acid impurities, possible impurities within this biopesticide. Identification of these organic materials that are present in the test substance were performed by comparing the mass spectra of the peaks to our NIST library match of the mass spectrum of each peak. As you can see here in our table, the far right column named match factor score is essentially a measure of how well the mass spectrum of the analyte peak matches the NIST library. It is a score out of 1,000. Now, in most of these fatty acids, we have labeled it the number and then a letter. For example, fatty acid 3 versus fatty acid 3A. And that is because the NIST library was able to produce 2 matches in which they may have been an isomer or some sort of chirality within the fatty acid. Now, it is important to note that most biological material components may not be identified in the NIST library. Therefore, we will use other type of methodology and analytical techniques, and also look at the literature to provide us with additional information on biopesticides for this type of material. Now, as an example of the complexity and nature of analyzing biopesticide, we have here a paper from Riaz Avar and her group, in which they have purchased 15 commercially available biopesticides, and they have used methodology ranging from HPLC to gas chromatography, GC, coupled with the MZ cloud and the NIST library to confirm suspected and or unknown compounds within this biopesticide. Overall, our findings of the components by our experiment were in agreement with the initial identification that this paper was able to produce us. As a result, for biopesticides, not only does the literature provide us with information, but also an ample amount of analysis have to be conducted to accurately determine each of its impurities and or fatty acids. Now, to recap, we have reviewed and completed steps 1, 2, and 3 of our Smithers 5-batch cycle for success. We have shown extensive data relating to how we screen unknown materials and how we determine approximate impurities of minor components and major components. The key part of this is through communications with our client, we now have a path forward to validate any active ingredients or impurities that are above 0.1% weight per weight. And these validations will be supported by our SANCO 3030-99 Revision 5 guideline. So let's take a look at what these guidelines impose. So these guidelines, we refer to them as enforcement methods. Now, the method validation parameters are performed to fulfill the testing guidelines under the European Commission SANCO 3030-99 Revision 5 set forth by the US EPA. The validation of the analytical method involves the following parameters: specificity, selectivity, detection, linearity, the limit of quantification, precision in highlight of a HORAT value, and our accuracy, which will be determining that either through marginal or total recovery. Now, the analytical, the reference standards for each of the batches that we have shown here will need to be thoroughly characterized prior to conducting this enforcement method. the analytical results of the active ingredient and all of the components present in quantities of more than 0.1% must be validated under the Sanko guideline. As a result, within the Sanko guideline, you also need to confirm the analyte. And we usually will employ NMR, FTIR, or three-dimensional spectra in which we have conducted our analysis via HPLC, UV, or liquid chromatography, mass spec, or even GCMS. And all of these parameters within our Sanko 3030-99 revision 5 guideline will meet the global regulatory prerequisite, whether you're producing your material and registering it within Africa, Asia, North and South Americas, Europe, or even Australia. Now, as I mentioned in the previous slide, Revision 4 and Revision 5 Sanko guideline are not significantly different in terms of the intended principles. However, Revision 5 has been published to remove a lot of the confusion on how we interpret the Revision 4 guideline, which was open to interpretation. Now, all of this data, which is vast and requires various instrumentations and technical expertise, are carried out here at Smithers, and we will help you develop these validated methods. There are some challenges when conducting a Sanko 3030 revision 99, revision 5 guidance on natural products or biopesticide or technical grade material. Some of these main challenges are listed here. First, we would like to ideally use one analytical method to quantify all the impurities. In the example that we have shown of the natural product, we were able to quantify 10 impurities within a single method. Next, the calibration range may vary due to the impurity concentrations. Therefore, a linear dynamic range will be imposed during the analysis. Next, the LOQ and high fortification concentrations to meet the Sanko guideline is extremely challenging. It's challenging because you're fortifying into the test matrix, which already has the impurity that exists. Next, the text matrix impact will especially impact biotechnical extracts, and we'll talk more about how we can relevate these challenges here. And lastly, when I mentioned that reference standards are not always available for natural products, therefore we may need to isolate those main impurities to then use that to validate our analyte of interest. And lastly, the precision and our accuracy have to be met depending on the concentration that we have found in the test matrix. Now, in order to mitigate some of these potential challenges, we have implemented a few strategies and solutions to ensure us to successfully conduct a Sanko compliant analytical method validation for the active ingredient and/or impurities. We will prepare a mixed analytical standard solution with all of the impurities. Our calibration range can be linear and/or we can use a 1/x weighted calibration range, which will ensure a larger dynamic range. Due to the variability of each analyte within a sample, we can run multiple dilutions of the fortified samples to capture each analyte to be quantified within our standard curve range. In terms of sample concentration, we can employ a direct aqueous technique and/or liquid extraction, or even a standard addition to accurately quantify the analyte. As I mentioned, without reference materials being available, we can isolate the impurity via fraction collection, or during the manufacturing process, we can also seek commercially available reference materials. Now, control samples containing the test matrix will provide us with a baseline concentration of that analyte, and therefore we'll know how much we need to fortify on top of that analyte in order for us to meet our LOQ and high-level concentrations. Now, as we discussed in the previous slides, the criteria for total closure for a 5-batch testing is at least 98% for the chemical components, and that has to be met based on the EPA registration of that test substance. As a result, other impurities and analysis within a 5-batch preliminary experiment are outlined herein. We can conduct a water content analysis via Carl-Fisher titration, and this will follow the CPAC method MT 30.1 and 30.6 to determine the percent water in your product. Sulfated ash analysis are used to determine the total inorganic impurities as a whole. However, if we do note that some of these inorganic materials have a concentration of less than 1 gram per kilogram, we may need to enforce ICP-MS for quantifying that metallic or inorganic material. Typically, we do not perform this for sulfated ash because they're always below 1 gram per kilogram, but you'll never know depending on the type of test material that we have to analyze. And lastly, the total organic carbon may be of use to our clients, especially for biological materials in which we have to determine both the organic aspect of that material. We can then proceed to conduct this experiment. Overall, the sum of these AI active ingredient impurities and these analyses can help meet their regulatory criteria to meet the EPA guidance. In putting all of these results together, a total closure of at least 98% was met for these 5 batches shown here. We have taken the summation of the active ingredient, the sulfated ash, moisture content, and the sum of all the impurities, and have determined that they are all meet the OCSSP guideline under the 830-1700 series. Lastly, if you have any questions, please scan the QR code shown here. We have additional resources on our Smithers website, specifically in the product chemistry portal, on how we can successfully help you determine any type of regulatory requirements that you may have on your product. With that, I would like to thank you all for being here today, and I will pass the mic to Solito for any Q&A questions. Thank you.
Yeah, folks, plenty of time left in your hour. We're at 37 after noon in the East. We have a few questions that have rolled in. First, Charles, we had a question from Elizabeth McDaniel. Are analyses performed under Sanko 3030 99 Rev 5 accepted by the EPA? And I guess we can expand that, right, to how different are those requirements in relation to, say, Europe or South America or any other global bodies moving forward?
Okay. First, thank you very much for your question, Elizabeth. The analysis performed under Sanko 3030-99 Revision 5 are acceptable by the EPA. So the EU guidance largely will follow the US guided in terms of our Sanko method. Now, if you're registering your product, let's say in South America, they may ask or seek additional confirmatory analysis. And this can span from a 3D analysis of the impurity of interest. They may seek for confirmation of the analyte by various techniques such as FTIR or even NMR. But overall, our Sanko 3030 method is applicable and accepted by the EPA in all of the vast regulatory bodies.
Right. So Charles, I work in a consulting company. So do you. One of the questions our collective clients are going to ask are, for a 5-batch testing on a small molecule TGAI versus a natural product. What is the turnaround time for conducting the analysis? And if you could reiterate some of those factors involved in that time difference, that'd be great.
Sure, Solito. That's a great question. So for a technical grade active ingredient with known impurities in which our client has disclosed that information to us, the turnaround time is usually 1 to 2 months. However, for a natural product with unknown impurities or major or minor components, the turnaround time for a 5-batch analysis can range from 3 to 6 months or even to a year. And that's really due to the complexity of that material and more importantly, how many impurities within that material we need to analyze and also validate under the SANCO 3030 Revision 9 guideline. But overall, if our client can provide us with additional information about their product, we'll be able to tune our analysis and therefore reduce the overall turnaround time for a 5-batch analysis.
As a follow-up question, Charles, I love that 5-step cycle that you presented earlier, and I think that visual is really important for our audience members to consider when they do scale up, when they do manufacture, and they reach decisions to that point. What do you suggest to companies with new technologies but don't necessarily know the anticipated impurities that they might encounter along the way?
That's a great question. So with that, I would suggest us to screen all the batches of interest. As I mentioned in these slides here, by screening these initial batches, we'll be able to identify those impurities of interest, and we'll let the client know whether these impurities are at a level in which it will be at or above that regulatory threshold. For materials that are, that don't have much information that our client cannot provide us, we can still employ various instrumentations to determine each of those impurity within that material.
Yeah, we had a follow-up question come in on the Q&A tool, which was, for a biological AI, specifically, what information could you possibly get to? What information would get the most mileage informationally to determine impurities in the 5-batch?
Thank you very much for your question. So for a biological active ingredient, we'll first like to know what that main active ingredient is in that biological entity. By knowing that, it will provide us with additional analysis in which we will be able to quantify that active ingredient in the biological material. The other 5-batch testing that may be of use is, of course, the Carl Fisher percent water. If this is a biological material that is to be used, let's say in a biological field, we'll like to know what type of percent water is within that material. Also, any impurities that are used or that may affect the active ingredient should also be identified. Of course, we can perform a sulfated ash experiment to determine any inorganics. Despite this being a biological material, we may not observe any inorganic material, but this is still useful for us to kind of have an overall profile of that product.
Okay, great. Is there anything special to bacillus strains that you've encountered? We have one of the questions in the Q&A. I might as well follow up that way.
To the best of my knowledge, I'm not fully versed in bacillus strands. However, I will have to get back to you about this biological question. Fortunately, I'm a chemist, so we really do not deal with much of a biological type of product when doing a 5-batch, but we have seen a rise in there. But I would say for for biological materials such as this, we can employ some targeted analysis. Fluorescence pops into my mind in determining the true active ingredient within that strain. We can also employ various GC or LCMS methods to determine concentrations of each of those biological entities. But overall, I think with this question here, let me do some more research and I'll get back to you on that.
Yeah, I guess this is an opportunity to let our audience know that for any of these questions we didn't get to, particularly for time or for depth, we can please expect some answers to these questions that come in the coming days. We will collect all the questions that were asked in the particularly in the Q&A section. So we had a question that came in straight from the chat, which we typically don't encourage, but
Sure.
does the 98%, according to Mark Shen, apply to biopesticides such as coming out of fermentation? And do the 5 lots need to come from commercial scale production? Could you clarify a few of those details?
Sure. So the 98% total closure was set by the EPA sort of as the complete characterization and identification of the active ingredients and its impurities. Typically for this product, for the biopesticide example that I've shown here, this was not a commercially available material. This was actually to be used in a fermentation process. As a result, it's really in terms of this here, I would definitely have to say that the biological aspect of it can be used in terms of analysis of a 5-batch. We can still use our typical analytical methods and procedures to identify any sort of fatty acids that are within the biological material. Overall, we will work with our client to see what exactly this material is being used for, so that we can then identify the proper methods and techniques that are needed for them to register their product.
Let's back up, Charles, to instrumentation. We have a question coming in anonymously about the 300 degree temperature in the GC analysis for biopesticides. So do you think there are chances of denaturing biological molecules or biologicals at that temperature, and what are the mitigation efforts and tactics involved in avoiding that situation?
Yes, thank you for your question. I definitely believe that at 300 degrees, we may be, denaturing some of the components within a biopesticide. For this analysis here, we used some of the conditions that we have found in the literature as a starting point. And then from there, we can tune our analysis. Of course, 300 degrees is fairly high for biopesticide analysis, but we can use liquid chromatography mass spec, in which we are now moving towards that type of analysis compared to a GCMS analysis. Therefore, we'll be able to then ensure that none of the components that are within the material is being denatured.
Great. From the Q&A tool, we have question from an anonymous attendee on your experience on the 5-batch analysis for metallic pesticides with ICP-MS. So I guess it would be in lieu of any further clarification, I would figure it would be small molecule actives with a metallic center.
Yes. So thank you for your question. One example that does pop in my head in terms of us, it wasn't quite a 5-batch analysis, but it was a C of A a certificate of analysis that we conducted in which there was a metal present in the material. Therefore, we did use ICP-MS to quantify that metal that was within the material. We have yet to conduct a 5-batch study in which we use ICP-MS to quantify any of the inorganic materials. But if that product does contain a metal, we can definitely use our instrumentation here at Smithers to ensure that we have proper quantification of the metal of interest within that product.
Right. Another question. We're bombing you here, Charles. From Jim McIntyre. What is your standard method to quantitate an unknown? A correlation factor of nearest known peak or a correlation factor of technical or area percent? I'm assuming as well internal standards might be employed here as well.
Yeah, so I thank you, Jim, for your question. Our standard method to quantitate an unknown will be if that material is GC compliant, we will utilize GCMS with the help of the NIST library match. In the example that I showed of the biopesticide, the NIST library match determines the match factor score, in which if it has a complete a correlation with the analyte peak and our NIST library, we get a higher match score. Now, that doesn't always mean that, that analyte is in fact what the NIST library match has identified. Therefore, we will employ a LC-MSMS analysis time of flight to accurately determine the mass and confirm the true identity of that analyte. So in all, in terms of our standard method for any unknown impurities, we will first make sure that we have GC methods in order for us to screen the material, and then use a more sophisticated method to confirm and identify that material within the product.
Right. So is the 5-batch requirement the same for proteins or nucleic acids? Probably some of the other
Yeah.
the RNA technologies, I guess, would be also in that. Go ahead, Charles.
Yeah, this is a great question, and thank you. I think right now we are seeing a rise in 5-batch requirement for these type of materials. Of course, the guideline doesn't really provide us with much help on the type of material in terms of biologics, but we have seen a rise in where 5-batch is needed for these type of materials. And once again, we will work with our client to determine the best path forward for identifying the active ingredient and its impurities within such a material.
Great. Okay. Two more questions here in the queue with still about 7 or 8 minutes left. Going from pilot scale operation to full commercial production, do you have to redo the 5 lots? I mean, I might even have an answer to that one.
Yeah. Yes. Overall, yes, you do have to. Just based on the guidance here provided by the EPA. I mean, going from a pilot scale to a full commercial production, you are producing more of the product, and in that case, you may need various labs to do that, and therefore that will be considered as a new or change in the manufacturing processes. And so you would have to redo a 5-batch analysis on those 5 lots that are being produced on a commercial level.
Mm-hmm. Yeah, from my perspective, and I've led clients on this journey as well. I've always thought that if you're scaling up by about 10x, you're going to end up with 10 squared the number of problems moving forward. So it's always good to, at least at the pilot scale, as part of the pilot scale to production scale due diligence, to at least monitor a few of these items that we already have discovered that are monitored by the 5-batch analysis, so that you can at least develop an understanding about any factors that you might that might give rise to a non-scalable process at the end of the day. So,
Yeah.
do you find any differences of how that gets enforced between EPA and Europe?
Yeah. So yeah, the guidance for that is very vague, but I will say this. If the EU guidance for 5-batch analysis follows some of the US sample methods and validations, then you should not have any problem in registering your product. Those two the sample guideline is can be used interchangeably between the two countries. So you will not have any problem in terms of registration.
Mhm. Question rolled in. Yeah, this actually kind of my last aside was kind of segues into this question. For the accuracy of active ingredients reproducibility, right? Limit of detection and quantification are not required under SANCO. However, the authority in South America requires a level of detection and level of quantification in the validation study. How would Smithers address a situation like this if following under the SANCO guideline? Let's assume that it's a botanical or an extract product. Thanks so much.
Thank you for your question. So in terms of how we will address LOD or LOQ quantification, that will be based on the sensitivity of that analyte that we're detecting, and also the sensitivity of our instrumentation. Usually for method validations, we would like to go as low in terms of our limit of detection that we can achieve. And then from there, we'll determine what our LOQ level will be. Our LOQ level is impacted by our test matrix. So whatever that initial concentration of your analyte is within that product, the SANCO guideline has set that limit plus the fortification of that impurity within the validated method. So our LOD will be determined based on the detection of our instrument, and then our LOQ concentration will be set based on the concentration of the initial analyte in the test. And we usually conduct that by having 5 representative control samples that have not been fortified in order to determine what the concentration of that impurity is. And then we will fortify on top of the test matrix at a concentration that we will expect will be the minimum within all the 5 batches of that sample. So that's kind of how we will navigate on how to validate a method that will consist and to meet our LOD and LOQ detections.
Okay, great. Do you use high resolution mass spec, and what mass accuracy do you achieve through that?
Oh, that's a great question. Thank you. We do use high resolution mass spec. Off the top of my mind, I believe our mass accuracy is plus or minus 0.0002 ms, but don't quote me on that value. But yes, we can achieve very, very high resolution analysis with our time of flight liquid chromatography mass spec detector. In terms of the value, I'm not too sure, but it's very, very low in terms of like PPT level.
Okay, great. One last question before we close out. Have you used surrogate standards to validate and quantify analytes that do not have a standard available?
We have used surrogate standards before, not specifically under the Sanko guideline, nor for a 5-batch, but we have used surrogate standards for Sante guidance analysis, in which we're validating the analyte and aqueous matrix. If let's say if our test material does for a 5-batch analysis, we'll need a surrogate. We can definitely identify and quantify that the analyte of interest based on the reference material from that surrogate.
Great, Charles. Thanks so much. And thank you to the audience as well. If we can, Charles, if we can move the slide up
Yes.
to close. Thanks. Thank you very much, folks, for your participation in this webinar. I would like to thank Charles Nyamekye for an excellent job, as well as the Smithers team, Eurofins Agroscience Services, our sponsor, and our webinar coordinator, Kyle Sawyer. I did have a correction on the student award travel deadlines. Those have passed at this point as of April 1st, and announcements for that will happen at a later date between now and May. For the questions that we did not get to, please expect answers to those questions to be posted in the next few days. You as an audience member may be asked to complete a follow-up survey after the webinar, and we look forward to your feedback based on that. Our next webinar is on May 15th at 12 noon in the East in the US, featuring Rory Mumford, who will speak on the non-target transformation product identification solutions for the 2023 EFSA drinking water guidance. And it looks like based on EPA actions, it's coming for us in the States as well. So it's going to be an important up and coming issue globally. So with that, thank you all for your attention, and we will see you all on May 15th. Have a good day.
Thank you. Thank you, everyone.